the very fundamental rules about development in human organisms. Hence, bacteria and worms and fruit flies and eventually mice are going to be extremely important in this regard. One of the most exciting concepts to me is the idea that we can use the Genome Initiative to essentially identify the molecular recognition units that are control units that regulate each gene. And I can see the time in the future when you can dial into a computer kidney and it'll print out 10,000 different genes that are expressed in the kidney. And you can print out a particular cell in the kidney and that'll cut out the subcell of genes that are expressed in that particular cell. That is, it has to be that there are molecular addresses that serve to coordinate the expression of these large sets of genes that are present in particular cells. So once we've identified these molecular area code addresses, we'll be in a wonderful position to use computers to make conjectures and then to go to real biology to test our ideas out. And it follows from all of this, as I said before, by the year 2000 or so, none of you will be cloning or sequencing human genes. That'll all be done, or much of it will be done and will be in the computer waiting for you to figure out clever things to do with it. Now, I've already pointed out why we should sequence mouse at the same time we sequence human to get these conserved regions that tell us about genes and regions of regulation and chromosome functions. But what I haven't pointed out is that proteins, it appears, are made up of a fundamental series of building blocks. They're called protein motifs. We've maybe identified 20 or so of them. My guess is there may be an alphabet of perhaps somewhere between 100 and 500. The Human Genome Initiative will give us all the genes, and by computer analysis that will give us all the motifs. And once we have the motifs, they'll be important in two ways. Number one, the motifs will be associated with particular structures, so they will be one fundamental building block to helping us predict the three-dimensional structure of proteins. And number two, particular motifs are associated with certain functions, and they will be one of the fundamental keys for us deciphering what unknown genes do, that is, looking for their corresponding protein motifs. But again, to return to the whole issue of the fact we'll have all the genes, we'll be able to translate to their protein structures, and I think in the same time scale as the Genome Initiative is done, we will decipher the rules for determining the three-dimensional structure of proteins using theoretical energy minimization calculations, using the ever-increasing library of three-dimensional structures, being able to carry out chemical and in vitro mutagenesis on particular structures, and the thing I just mentioned, these protein motifs that we've talked about. But once we can predict the three-dimensional structure of proteins, we are a long ways from being able to figure out what they do. That is a problem essentially unsolved for any protein today. Even hemoglobin, the best studied protein, still has advocates arguing over alternative ways for executing some of its functions. So one of the challenges of the future is to go from three-dimensional structure to proteins. The other point that I would make is we are developing automated techniques for 2D gel analysis. So you can use these two-dimensional fingerprints of complex mixtures of 5,000 proteins. You can digitize the information. You can look at those patterns of proteins in cells that are normal and have been neoplastically transformed. And then you can look at the 10 or 20 or 100 spots that are different in those two cells. We could micro-sequence those proteins. We could put those sequences into the computer and instantaneously identify the 100 genes that are different in a cancer cell from a normal cell, and then go straight to questions of why have those proteins varied quantitatively or qualitatively. So this will be the subtractive analysis coupling 2D gels together with the Genome Initiative will be an extremely powerful tool in the future. So I've given you only a few examples of how biology is going to change, but the change will be profound indeed. How about the benefits to medicine? Well, again, if you've got all 100,000 genes, you're not only going to have wonderful potential therapeutic reagents, but you're going to open up the whole world of DNA diagnostics. And let me just say a word about DNA diagnostics, because that is, I think, one of the most profound changes that's going to come to medicine in the next 15 years. So again, DNA diagnostics is looking between a probe and a target chromosome for molecular mismatches. And I've already talked about this technique of DNA ligation to be able to detect the variations that you see in humans. So you can use this technique as a very powerful tool for DNA diagnostics. And in fact, we've used it to study a whole series of human genetic diseases very, very effectively over the last 6 months or so. Now, to give you an idea of the power of what this DNA diagnostic technique can do, is it really is going to allow you to identify genetic mutations that are stable. That is, for this technique to work, you have to know the mutation, so you can pick out common genetic diseases in large populations. And indeed, we're setting up to do very large-scale screening for cystic fibrosis in this particular regard. Another area that is particularly important is to identify genes that predispose you to disease, whether it be the cardiovascular system, whether it be autoimmune, whether it be particular types of heart disease or nervous diseases of the central nervous system or what have you. And I'll talk more about disease predisposition in just a moment. This can be used to look at dominant and recessive oncogenes and to study cancer in various ways. DNA diagnostics is really going to revolutionize the diagnosis of infectious diseases. It's going to be rapid, it's going to be exquisitely specific, and it's going to be able to give you the potential to subclassify in ways that there is no possible or conceivable way that the more biological approaches could do it. And this is clearly going to be the way forensics will be carried out in the future. Now, one of the issues that comes up is if you can do DNA diagnostics and genetic diseases or genetic predispositions, is that a good thing? And I think to a first approximation, the answer is no, unless you can do something about what you've diagnosed. And of course, the approaches will be the avoidance of environmental dangers, molecular pharmacology. As we figure out how all these systems and their genes work, we'll be able to design rational therapies. Immunotherapy, I talked about this afternoon. Gene replacement. The first gene replacement experiment has already been done. Human bone marrow cells for an enzyme that causes a particular immune deficiency disease. But what I would really emphasize for the future is I think in 15 years we will identify 100 genes that predispose us to disease. And we will be able then to take newborns, to analyze their DNA, to feed that information into a computer, and to say unequivocally that this child is predisposed to these 4 diseases, and more than that, this is what the physician should do to circumvent the potentiality of those diseases. So what DNA diagnostics will be able to do is move medicine into a preventive mode, away from this reactive mode where we try and make sick people well. There will be enormous cost savings. For example, rheumatoid arthritis costs American society somewhere between $1 billion and $1.5 billion. Suppose we had diagnostic techniques that could essentially circumvent that single autoimmune disease. Again, the savings to society would be absolutely enormous. I talked this afternoon about techniques whereby we can use a study of the receptors for the immune system to predict whether or not you'll be susceptible to particular autoimmune diseases, and use that knowledge then to avoid these autoimmune diseases. I can also point out to you that in the future we'll be able to map genes probably in a matter of weeks. And let me give you the counterpoint to this. The gene for Huntington's disease was localized generally almost 7 years ago now to the short arm of chromosome 14, and they still haven't gotten any closer to it. With the entire sequence of the human genome, we can use computers to look at the sequence and the region of the gene. We'll identify all the genes. Well, then there are a variety of strategies you can use to determine which gene is likely to cause the particular disease trait, and then you can check the hypothesis you've made against the genes that are present in patients with the disease. Now, to drive home what an effect this is going to have in cost savings, Francis Collins estimated that it costs somewhere between $150 and $200 million to isolate the gene for cystic fibrosis. If we had this sequence of the human genome, I would estimate we could isolate virtually any single gene for well under $100,000. So the cost saving is absolutely incredible in this regard. It's also quite clear that with our ability to put genes in just exactly where we'd like to put them, this is called homologous recombination, there is going to be then a world of making models in mice of human diseases wherein we can study physiology. And of course, there are limitations in mice because they behave sometimes differently than humans, but to a first approximation, these mouse models should be marvelous for working out the tools of molecular pharmacology. And finally, I would say in the future, therapeutics are essentially going to center around our ability to look at a protein sequence, to determine its three-dimensional structure, to determine a complement to that three-dimensional structure, and then to make either a protein or a very small organic molecule that can recognize any part of the protein you'd like to serve as a target, to which could be coupled therapeutic reagents that could kill, in this case, a cancer cell, for example. So protein engineering, I think, is really going to revolutionize what we can do in human medicine in the future. Now, there are naysayers for the Human Genome Initiative, and these are the major categories of objections that have been raised. Some have argued that it's bad science, and I think that really comes from not understanding what the Genome Initiative is all about. And it's about two things: technology development and providing an infrastructure, which is the computerized sequence of the human genome. It isn't science, it's providing the tools to do science, and it's providing tools that are going to revolutionize how we do science. They've argued that it's big science, and that again is a fundamental misunderstanding. What the Genome Initiative demands is interdisciplinary science. But the group we have at Caltech, we don't have more than 2 or 3 people working on computers or working on nucleic acid chemistry or working on large-scale sequencing. But what we do need to put together is all these groups, so their combined expertises can network to generate the kind of tools we're going to need for the science of tomorrow and solving the Genome Initiative. And this is network science, interdisciplinary science. It isn't big science in the sense of the super collider. That it's an inappropriate training ground for young scientists, I think, again, is a lack of a fundamental misunderstanding, because I think the people that we're going to train that are going to know biology and these other technologies, frankly, are going to be the real leaders in biology of tomorrow. And that is exactly the kind of people that we should train, and I think the Genome Initiative provides a unique opportunity to provide them with these interdisciplinary tools that the future is going to demand. The real objection to the Human Genome Initiative is this last issue. And the unfortunate truth is the Genome Initiative makes up 1% of the budget for molecular biology, and if you erased it, it wouldn't help general biology at all. So there is a crisis in funding in the United States today in science, and that's something we all have to fight against. But I think the Genome Initiative is not going to provide an answer to that real crisis, which has come about because of a lot of very poorly thought out decisions at NIH about how they're going to carry out long-scale funding for science. There are a lot of ethical problems that can come from various issues in DNA diagnostics, for example. If you can determine for individuals their predisposition to disease, there are fundamental questions about confidentiality. That is, does an insurance company or your employer, your future employer, have right to access to that information? And I would say unequivocally no. We have to set up safeguards so that information is held between you and your doctor. The question of prenatal diagnosis, DNA diagnostics, is going to let us analyze any human genetic disease. You can make a rational decision about therapeutic abortion. And of course, this is an area of enormous collision at this point in time in society. The long-term implications of being able to engineer human beings. Let me say that the engineering in our lifetime is basically going to be single gene, single trait engineering. and they don't touch the fundamental properties of humanness, intelligence, emotional stability, physical attractiveness. These are all complicated multigenic traits that are off in the future. What is interesting is the Genome Initiative is the first initiative that has put 5% of its budget into considering these kinds of questions, trying to make a step forward in their resolution for society. So I think you can see then how the Genome Initiative is going to give us the tools to approach this complicated problem of the development of the human organism, starting as a single cell and going through this area code array of differentiation. I think you can see how it is going to dramatically alter medicine in the future, shifting it from this reactive mode to a much more preventive mode. And what it is also going to do is give us very powerful tools for looking uniquely at each individual and designing therapies that are specific for their physiology and their disease, something we can't begin to do at this point in time. And of course, as we understand how the networks of genes for the cardiovascular system or the immune system or the nervous system begin to work, we can start to approach on a rational basis the diseases that arise as a consequence of genetic defects in these particular systems. So I think you can begin to understand then that the unique feature of the Genome Project is that it really is closed-ended, that it will be possible to do at least much of the human genome sequence. The results will be interesting, incredibly useful, striking technologies will merge, and a point I haven't really talked about is the problem is international in its dimensionality, and a real challenge is going to be working together with other countries of the world. So in closing, let me thank my many colleagues in this interdisciplinary effort we have at Caltech that have carried out the development that has made possible a lecture like this. And you can see that it isn't very many people, but the interdisciplinary nature and the interactions of their talents can really have quite remarkable effects. And I think this is one area of enormous opportunity for biology in the future. Thank you.
Dr. Hood, and that is a reception here, right in the back corner here after question and answer. Since we're running a little late, I'll allow about 10 minutes for questions, and if you want to talk to further, you need to discuss.
Yes.
Several companies have been doing work very similar to the work you described. And I wonder if you could tell us about the relationship between the government-funded Human Genome Initiative and these private enterprises with regard to whether they have copyright and patent.
Well, that's really a good question. Let me tell you about my philosophy of the difference between academic and industrial research. We take the risks. We try to develop the new technologies. We develop the prototypes, then the industry has to go on and package. And under this rubric, the technologies that we developed have, for example, all been patented by Caltech. So if a company then takes them for commercial distribution, they pay to Caltech royalties that go to the institute and go back to the laboratory that's made these discoveries. Now, with regard to the whole question of sequencing the human genome itself, the Genome Initiative is going to make everything public. Nothing is going to be copyrightable. It's going to be published. In fact, one of the real imperatives for the Human Genome Initiative, when you write a grant for large-scale DNA sequencing, you make a formal commitment about how you are going to make public your sequences. And there is enormous pressure to make them public before you ever write papers on them so that they are in the database and they are available to anybody who might want to do anything with them. Now, having said that, when companies clone genes and sequence genes, they do have the right to patent these things, and no one can alter that, although they are going to find it more and more difficult to do that as this large-scale sequencing effort really swings into full operation. But quite frankly, that isn't going to happen probably for 10 years just because it's too costly to sequence now. To give you an idea, the naive labs that do DNA sequencing, or I should say the unsophisticated labs that do DNA sequencing, when you calculate in all the costs, probably it comes to somewhere between $20 and $40 per base pair. Now, the large-scale labs that have really optimized things can push this cost down to $2 to $3 a base pair. The objective of the Genome Initiative is to push it down to 5 to 10 cents a base pair. And I think that's certainly going to be doable over the next 10 years. But all of the instrumentation I talked about here we've developed at Caltech has been commercialized, and in all cases, Caltech and the government, of course, have the patents and primary control over them. Other questions? Yes.
Yes, I was wondering about the lifetime between the identification of genetic diseases or susceptible diseases and the actual development of gene therapy.
Right.
Therapeutic approaches. And one of my concerns is during that lifetime, what are the legal ramifications for somebody with an identifiable disease who chooses not to avoid a certain behavior or situation? Are they liable for their disease costs and things like that?
Well, I'll tell you what my policy would be. Look, diagnosis is always going to precede therapy. I mean, that's a given. I would argue we should never, ever do diagnostics except for one of two justifications. One would be that we have the therapeutics that can complement it, or the second would be in some cases for the question of therapeutic abortion. I think that is a legitimate kind of thing. But beyond that, for example, if you come up from a family that has Huntington's disease, I think you have to think very carefully about whether you ever want to have that test done on you, because it's a hell of a thing to know. I mean, it's great if you don't have the chromosome. If you do have the chromosome, then you have to wait until you're 30 or 35 or 40 or 45, and you're going to go through just a horrible neurodegenerative process. So my own general policy would be you never do DNA diagnostics unless you have a therapeutic approach to it, or the question of prenatal abortion is raised as an issue.
And what do you foresee as lifetime? Gene therapy just was initiated in the last 2 weeks. Do you see that as
How rapidly will it come?
Yeah, with the genome
Won't begin to keep pace with the Genome Initiative. I think we don't know how long it's going to take to get generalized gene therapy, and by that I mean having vectors that can deliver good genes to any target organism in a human. That just seems like it's a long ways off, although we can think about great theoretical ways to do it. It's proven very difficult. But I think once the general problem is solved, then things will be done very, very quickly. And maybe it'll take us 2 years, and maybe it'll take us 5 years, and maybe it'll take us 10 years. But I think once we do, then I think we'll have enormous capacity to target genes to wherever we'd like to target them. And I think, frankly, in the end, it may well be done using these solutions of molecular complementarity if you can identify in the target organ a unique protein on the cell surface. You can make its complement, and you can use that then to target viruses or whatever it is to that. I think those are going to be some of the strategies that will be generally successful in the future, but it's going to take a while. So now, the other thing I guess I would point out is, will diagnostics really outstrip therapeutics? I guess in a general sense the answer is no, because you have to realize we're not going to do very much human sequencing for the next 10 years. I mean, my guess is by the end of the first 5-year period, I think we'll have done 0.2 or 0.3%. By the end of the second 5-year period, I think we'll be very lucky to be to 1%, okay? But then I think it'll explode after that. And if that is the time scale of genetic engineering, then it could happen right in parallel. Yes? What kind of DNA? Non-coding DNA, right. 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 Two good questions. First, with regard to the issue of junk DNA, or the fact that maybe 97% of the DNA is non-coding, my own feeling in this is, number one, there is a lot of information in that other DNA. Number two, we don't know where the information is, and the easiest way to find where the information is to actually carry out the sequence. Number three, we don't know what fraction will be informational in some way, but I gave you examples of some of the other things that will be buried in non-coding DNA. Regulatory information can be hundreds of kilobases from particular gene families. Certainly there can be a lot of information that's conserved that relates to chromosomal functions, such as this compaction and so forth. So I think it's going to be very important to do the entire sequence to find the information. We also aren't going to be able to know where the genes are. So the only way to get all the genes is to make sure you've sequenced everything. And the final point I'd make is in 10 years, we're going to be able to sequence 10 million base pairs a day, or 100 million base pairs a day. So the problem is going to be very different in its dimensionality than it is today. So I think you have to keep that whole issue in mind. Now, the issue of repetitive DNAs is really a good one. My own feeling is the centromeric repetitive DNAs, or the very, very large block of very similar repetitive DNAs, won't be sequenced. I mean, what I think you'll do is you can use clever techniques to measure very precisely their size, and then you'll assign a general repeat sequence to that, and you won't worry about it. The other interspersed repeats, such as allos and things like that, don't present serious technical problems for the sequencing. Now, they may present really serious technical problems for cloning. That is, we know there are human clones that are extremely unstable, and we may have to devise either very clever cloning techniques for stabilizing these intrinsically unstable clones, or what I think is much more likely, we're going to develop very, very powerful long-scale PCR technology so we can make transcripts right directly off the chromosomes and sequence them, and we don't have to clone things and worry about those kind of instabilities. But those are my points of view. So the repetitive DNAs, certainly at the centromeres, you're not going to bother to do those. You'll just make general measurements of sizes. Yes?
We hear a very different number about the cost of the project, particularly the percentage of the research budget that's going to go to this initiative. And I wonder if you comment about how way you make the 1%.
Well, I get them from NIH. I mean, the budget at its most is going to go up to 2% overall. Now, where the numbers could differ is what you include in the numerator, in the denominator, okay? So, what is molecular biology? But I can say this. I can say NIH in a general sense spent $7 billion last year on research. The Genome Initiative for NIH was about $50 million. That's an incredibly small fraction. I can say for AIDS, they spent more than 30 times as much on the Genome Initiative. And I would argue from all the things I've said here, this is a hell of a good thing to do for molecular biology. And if we can invest 1% or 2% or 5% in it, then I think intellectually we are an incredibly impoverished nation. So, I mean, that's how I answer these people. I mean, you can come up with different denominators, but it really doesn't matter. Is it 5% or is it 1%? It is a small fraction.
We can take one more question, and then you'll have to take your chance for 7.
Okay. Well, you've asked about 3 different questions. I mean, the first one is the most interesting one. How accurate can you sequence DNA? And the answer is we don't have any idea, because there aren't any very good objective measures to characterize it back against. But we're trying to devise tests to do that. Now, the questions of how you assemble these little strings into big strings, there you have to build algorithms that can tolerate 20% error rates. And they are what make it so incredibly computationally intense. But there actually are people that have done very clever things in that regard. One of the interesting conclusions that I've drawn is that the biological problems posed to the computational people are going to let them get at very, very fundamental issues in computational science. And they're going to lead them to completely new ways of thinking about computer science. And to give you an example, Ken Kennedy is director of the Science and Technology Center for Parallel Processing at Rice. And a couple of years ago, we started talking about this DNA assembly problem, and we've done some things together on it. And he told me recently that he's working now with Intel with their latest super chip. And he says it's interesting, but the most fundamental part of the new algorithm they're developing is essentially the DNA assembly problem. So there are going to be really interesting offshoots from the computational problems we'll solve for biology, I think in many other disciplines. The chip we've developed, this chip, is going to have an incredible impact, I think, on geophysics, because it is a chip that can search through seismic patterns. You know, when you shoot off a charge of dynamite, you get an incredible array of patterns from different points that you have to analyze. And this chip is designed to do a pattern search that is made to order for that kind of thing. And the global environment business, looking down from satellites at the Earth and generating enormous arrays of information, again, some of the pattern search problems fall into these chips that we've talked about here. So there's a very interesting commonality in that regard.
Thank you, Dr.
Well, you guys put it all together. That was pretty impressive.
And if you want a copy of it, you can just write down your request for copies.
Okay. Okay.
We're in the neighborhood. Had to stop at Carver at 9:30, so I couldn't.